Thank you very much. I want to first start by thanking the organizers for giving me the opportunity to talk about this work. It's the first time that I'm coming to this workshop, and so I hope that what we do is in some way important for drug discovery, but I guess we can discuss about that. So I'm interested in general in mechanisms, and in those respects, then obtaining the conformational free energy is much better than having access to a single structure. And so we've been very interested in developing methods to do that more efficiently. And this talk will be about an application to the Calmodulin conformational ensemble, but the methodological developments should be useful for any other application, basically. So more practically, it's been known for a while, actually, that we can get additional information for drug discovery by considering conformational ensembles rather than single structures. And this is an example taken for G-protein coupled receptors where ensemble docking actually performs better than docking to a single structure. But the question of how to obtain the conformational ensemble remains. So I think in this case, you would run MD simulation and obtain a conformational ensemble, but much more relevant would be to obtain the actual conformational ensemble that the protein samples during its functional cycle. And this is not a trivial thing to do at all. But before I go into the details of how we do this, I just want to introduce the actual object that we're interested in. So it's Calmodulin. Calmodulin is a completely ubiquitous protein. We have in humans 3 replicates of the gene, and if one is dysfunctional, the organism is not does not live. So the way it works is it binds calciums, and then it binds to a variety of target proteins that were originally not calcium sensitive, and that then become calcium sensitive upon Calmodulin binding. So this is just 3 examples of pathways. And I come from a background of membrane proteins, specifically ion channels. And in those contexts, Calmodulin regulates indirectly or directly pretty much any ion channel. So this is why we've been interested in it. And the idea is that then characterizing the Calmodulin conformational ensemble can help to understand how it binds and how it regulates the function of membrane proteins. So if you look at a static view, and if you look at structures that are known of Calmodulin in the absence of calcium, in the apoform, or in the presence of calcium, you can see that seemingly a large conformational rearrangement happens upon calcium binding. In the presence of calcium, the structure is quite rigid. So you can see Calmodulin is made of 2 lobes. Each of them contain 2 EF hands that bind calcium in black. And in the presence of calcium, these 2 lobes are pretty open. They expose hydrophobic residues, and that allows them to bind to hydrophobic targets and to then regulate the function of the protein. In the absence of calcium, you can see that the structure is quite different. It's much less rigid, and the lobes are collapsed onto themselves. And in that case, it's much more difficult to bind a target protein. But of course, this is just a static view upon resolution of a crystal structure. And in fact, the protein actually samples quite a lot, a large landscape when it carries out its function. And so molecular dynamics, in principle, is well suited to explore the conformational ensemble because it allows an atomistic resolution directly, which most experimental techniques don't. But the problem, as we've heard already many times, is that it usually, if the free energy landscape underlying the functional behavior of the protein has high free energy barriers, then if you start a simulation in a metastable state, then you will stay stuck there for a very long time. And there are many techniques that allow to overcome free energy barriers. So this is adapted from Alessandro Laio's webpage and illustrating the way metadynamics works. But we heard this morning about ABF or about umbrella sampling and other techniques that allow to overcome free energy barriers and then sample a much larger part of the conformational landscape. Many of these techniques actually, if you modify the Hamiltonian in a controlled way, you will be able to recover the free energy landscape that was underlying the conformational landscape. But the problem of many techniques is that you need to have access to a collective variable or a degree of freedom that is the slow one or the ensemble of slow ones that will, along which to enhance the sampling. And this was mentioned already a few times, and actually yesterday Pratyush showed us that there are now ways to discover these collective variables in a data-driven way. But still, if we can, we'd like to use free energy enhanced sampling methods that don't rely on having access to a collective variable, especially in the case of Calmodulin, where you've seen the conformational changes far from trivial and highly nonlinear. So we've been interested in using these temperature-based replica exchange sampling schemes. And basically the idea here is that you run multiple simulations in parallel, that you start at different temperatures, and then you attempt exchanges between the different temperatures, and you accept those with a metropolis criterion or reject them. And this allows to propagate what is going on at higher temperatures back to the lower temperature. So basically you're taking advantage of the temperature fluctuations to collect statistics at the low temperature. And this enables to overcome free energy barriers. So for the conformational ensemble of Calmodulin that we were interested in, we basically wanted to compare free molecular dynamic simulations, just regular MD, temperature-based replica exchange that I just explained, and one variant, temperature-based replica exchange solute tempering, which I'll explain in a minute, which is basically a variant of temperature-based replica exchange, but that allows, that has a lower computational cost. And so the idea is that these methods have been around quite a long time, but they haven't really been contrasted, and the computational cost associated with these three methods on such a complex problem has not been fully evaluated. But before we did this, we stumbled upon a problem that is sometimes, or maybe much of the time, overlooked. And that is having access to a method that estimates probability densities, which are related to free energies, as everybody knows, through the Boltzmann relationship. And the idea here is that by default, I believe in the field of molecular simulations, what is done is basically histogramming. So the way histogramming works is you select the collective variable along which you want to estimate the probability distribution. You discretize it, and basically you calculate the value of the collective variable for each individual sample that you have from molecular dynamics, and then you increment the bin in which you find the system. And you get, for an idealized density like this, what you get is this kind of estimate. And immediately what pops up to most people is that intuitively you can tell that this is not a good representation of the data. And the reason for this is that biomolecules live on a smooth landscape. They don't live on a discrete landscape. And so when you see this, you can immediately tell that this is not a representation of reality. And then, more importantly, discretizing poses several problems because the final probability distribution will depend quite a lot on the size of the bin that you use. If you have too many small bins, you end up with a lot of empty bins. And also, to some extent, the offset and the place where you set the grid can pose problems. So we thought that we would look into other probability density estimators, since they are available and widely used in other domains of science, actually. So I mentioned histogramming before, and traditionally, Gaussian-based methods, these three listed here, kernel density estimator, Gaussian mixtures on a fixed grid, and Gaussian mixture models are used, and also k-nearest neighbors, which basically estimates the density from the inverse distance to the k-nearest neighbor. So the further the k-nearest neighbor, the lower the density, are all quite used to estimate probability densities in other domains. And these can all actually be formulated as a sum of basis functions that all have the same shape. So I already mentioned histogramming quite a lot, just to explain the functional form here. You have the probability density, which will depend on a set of parameters that is a sum of indicator functions. So indicator functions are if the point is found in between in the bin in question, then the indicator function takes number 1 and it takes the number 0 outside, and then you have normalization factors. And as I mentioned, the counting basically is optimizing the amplitude of these indicator functions given the data, while the parameters that are spurious and that cause problem, and something should be done about this, are the offset and the size of the bin or the number of bins. If you look at these other functional forms, they basically have the same type of sum of basis set, except KNN is actually a sum of delta functions. And there you basically the k in the k nearest neighbor should be validated, because it is a spurious parameter, as is the case of the offset and the size of the bin in the case of histogramming. And then in the case of these Gaussian, case of these Gaussian-based estimators, basically the density is a sum of Gaussian functions with variance sigma and amplitude a here. The difference between these three methods is then the way the parameters to be optimized or not are set. So in KD, basically, you will put a Gaussian at every point that you consider. And the only parameter that needs to be estimated is the width or the variance of the Gaussian. So the number of Gaussian is fixed because it is equal to the number of points that you're estimating the density for. In the case of GM grid, basically Gaussian mixture on a grid is a continuous version of histogramming, and you can recognize offset and a spacing between points as meta parameters, while the amplitudes need to be optimized. And in Gaussian mixture models, it's the opposite, actually, where the parameter that you're optimizing are going to be amplitudes, variance, and the place of the Gaussians, whereas you are varying over the number of Gaussians. So this is going to be a meta parameter that we need to find. So brief description of all the probability density estimators. And before we went back to the problem of Calmodulin, we basically evaluated the performance of these estimators on a toy model, for which you can see the probability distribution here in black and the corresponding free energy here in gray. So we picked this specific form because it kind of looks like a double well potential, but you do have uneven features that make the density estimation a bit more complicated. And basically, we evaluate two things: the accuracy, which is the deviation of the estimation from the true profile, And we estimate the stability by estimating the standard error for each estimator. And to estimate the parameters, what we did is to use a Bayesian approach. And this is basically, since we're trying to find the density model that best describes the data, we flip it on its head, and we're basically going to try to estimate the probability of the model given the data, assuming that the probability of the model itself is uniform. And the way we do this in practice is written here. So we're going to estimate the likelihood functional here of the trajectories given a probability density estimate. And we assume that all the points in the trajectories are independent, which is a strong assumption. But it is also present in histogramming, and we take care of taking independent samples in our simulations by decorrelating the samples we use. And to do this, basically, we use maximum likelihood. So we maximize this likelihood functional that I was mentioning through, for fixed meta parameters, we estimate the variable parameters, which are mostly the amplitudes, subject to normalization and positivity. And we solve this iteratively using expectation maximization. So these are the initial results that come from using a fixed set of parameters, 20 basis functions, or in the case of k nearest neighbors, k equals 7. And on these plots, you can see in gray, the true free energy profile, and in color, the estimate that comes from this maximum likelihood estimation I was just mentioning. And we sample from this probability density 10 trajectories of 200 points each. And I think it's difficult to see, but here we're listing the mean true error and the estimated standard error. And basically, these two estimators that are based on Gaussian functions, as I said, perform better to estimate the true free energy than the histogram and the KNN. Histogramming, as you can see here, performs particularly badly in the areas where you have few points because it's a high free energy region. And the standard error here that is shown with the dashed lines is huge in those regions. When we reduce the number of points that we are drawing from the trajectory from 10 times 200 to 10 times 100, we actually see that these estimators, KD and Gaussian mixture on a grid, perform worse, but histogramming really starts performing badly. And this is what I was saying when I was saying that when you have too many grid points, too many bins, basically the estimator performs really bad. But this was only in the case of one set of meta parameter. I mentioned 20 basis sets. And the idea here is to avoid overfitting by using cross-validation. And this is done to find the best set of meta parameter. So I explained this estimation just before. Adding in the cross-validation looks like this. So you start from the data, you split it into a training set and a validation set. With the training set, you estimate the parameters, and you can calculate the profile using each of these estimators. And then you estimate the likelihood on the validation data, which allows you to find the set of best meta parameters. And then using this set of best meta parameters, you will estimate the probability density of the entire data to get the profile. And these are the results summarized very quickly. For the best profile, given the best set of parameters for the 5 different estimators. Here on these plots, you have the standard error for each estimator, the true error to the free energy and the true error to the probability density, as well as the log likelihood. And you can see that along this axis, basically, you are doing better as you move to the continuous estimators and as you move to Gaussian mixture models on all counts. Especially interesting is the predictive power of the Gaussian mixture model. This is also a good occasion to show you that any estimator, even the one that performs the best, is actually still a model. You can see that these small features in the middle of the profile are never reproduced by any type of method. So you can always estimate probabilities or free energies, but remember that this will always be a model of reality. So with this method, then we can go back to Calmodulin and to compare these MD to temperature replica exchange and temperature replica exchange solute tempering. And basically what we did is we compared what was comparable. So we generated 5 microseconds worth of data for each of these methods. For MD, it was 7 independent simulations, but for temperature-replica exchange, then since we have 25 replicas at different temperatures, each replica is going to be 200 nanoseconds, so the whole computational cost is the same, but the length of the trajectory is much smaller. In the case of replica exchange solute tempering, I mentioned briefly before, what is done is that only the solute is heated as opposed to the entire system. And this allows to span a temperature range that is much higher than in the case of replica exchange MD. And this then allows to overcome free energy barriers that would be much higher. But still, we are considering a total of 5 microseconds to be able to compare. And in both of these replica exchange simulations, the density is estimated at the lower temperature. So the aim of the swaps is only to propagate conformations that are accessible at higher temperature. We are using, as I mentioned, a method that is collective variable free, but still when we project the free energy landscape, we need to project it onto a set of collective variables. And here we picked 2 collective variables, a local one that is based on the similarity of dihedral angles, of backbone dihedral angles in the holo structure, and a global CV that is based on the distribution of reciprocal interatomic distances also to the holo ensemble, or also to the holo structure. And these are the resulting free energy profiles. So using this robust free energy estimator I mentioned, Gaussian mixture model with cross-validation. So the comparison is between MD in purple, temperature-based replica exchange, MD in blue, and solute tempering in green. And as you can see, in the case of no calcium bound here in the bottom, the profiles look somewhat similar. But in the case of the holo ensemble, the profiles estimated along the local collective variable or the global collective variable both look very different. And this indicates that the conformational ensemble that we have access to using these 3 methods are completely different. And likely it also means that the profiles that I'm presenting here are not converged for any of the method. You can see that, by the way, the standard error that is represented by the dashed line is pretty big everywhere, except in the minima close to the structure. To have a better view of this, we basically look at the probability distribution or the free energy along these 2 collective variables. So the global one and the local one on this axis. And these plots show these free energies estimated with our estimator for MD, temperature-based replica exchange, and solute tempering in the presence of calcium and in the absence of calcium. And as I was just saying on the slide before, these methods allow to sample free energy landscapes that are very different, especially in the case of holo. And I think this is even more visible on these type of plots. So basically, in the presence of MD, like I was saying at the beginning of the talk, the system stays really stuck where it started in, whereas replica exchange MD allows to explore a bit more of the landscape, and replica exchange solute tempering allows to sample even more of the landscape. And this is because the temperature range that is accessible is much higher. And this is compatible with the free energy landscape that is found here, since you can see that there are pretty large free energy barriers that separate these different minimas. So for example, this barrier is of the order of 5 or 6 kcal per mole. In the case of the APO landscape, the situation is quite different, and the sampling of the space is actually quite large, even in the presence, even when you use regular molecular dynamics. And this is because this landscape is pretty much flat and featureless. And this means that having a larger, longer simulations actually allow you to sample more extensively the landscape. You can see here that solute tempering, that would allow to overcome barriers, but that is limited in terms of the length of the simulations, does not allow to sample this region of the space, for example. So this is then difficult to extrapolate, because basically if you don't know what type of landscape you have to begin with, it's a bit difficult to know which type of simulations to use. And this came a bit as a surprise to me, because I was expecting that enhanced sampling free energy method would be more useful for any case, but it really isn't the case if you have a diffusive free energy landscape. And to finish, I just want to highlight again the effect of this estimator of the free energy on these two-dimensional free energy landscape. So this is the holo ensemble simulated with temperature replica exchange solute tempering. It's basically this plot here, but it's a comparison between the GMM, the GM grid, both with cross-validation and basic histogramming. And you can see that the features are basically the same. So you can find minimas in the same place, which is reassuring. But really, in the presence of histogramming, you have no notion of the height of the barriers, because these regions are sampled so scarcely that you have no data there. But having the Gaussian mixture actually allows you to infer the probability density also in the regions where you don't have a lot of data points, or even no data points at all. Another thing, so with these methods, basically you can characterize transition paths, both where they are and also the height of the barriers. I was also showing you that GM on a grid and GMM were performing both quite well in terms of the toy model that we used. And this is to highlight that these two models perform equally well, but they look quite different on the two things. So just remember that everything is a model, basically, even if you have a good, robust, and predictive method. So to conclude, this didn't work, so I have no idea how am I doing. So to conclude, the summary is that with a Gaussian mixture model with cross-validation, we can estimate quite accurately and robustly probability densities and free energy landscapes. And this is applicable to any problem that you may have if you have an ensemble of molecular dynamic simulation trajectories. The code is available if you would like to use it. And on the specific question of Calmodulin, we can say that temperature-based enhanced sampling methods only perform well on the rugged free energy landscape of the calcium-bound Calmodulin, while it's not the case in the calcium-free Calmodulin. And this actually has biological implications, because we know from our other investigations that calcium-bound Calmodulin adopts more of a conformational selection type of mechanism, whereas Calmodulin is much more flexible. And so the free energy landscape supports this difference in the binding modes of these two types of Calmodulin. We are thinking of making this method locally adaptive, because for the moment, this cross-validation only works on the whole landscape. This I just mentioned. Check out the paper if you're interested. And basically, we are also now modeling the binding mechanism directly using this kind of method. So with this, I just want to thank or acknowledge Annie Westerlund, who's a PhD student in my group, and she did all this work. It's been a very fruitful collaboration with Christian Blau, and I also want to acknowledge Tyler Harpole for discussions and for simulating some of those ensembles. And with this, I want to thank you very much for your attention.
I'm going to have the next speaker come down and start to set up. Questions for Lucie? Yes, Levi.
So for the able structure, you're using a profile from MD as a reference. So how do you know that's better than others?
Well, I didn't get it.
So for the APO structure, you're using the Affinity profile, using the MD as a reference, right? So how do you know that's more accurate than the others?
Okay, yeah, so you're saying if we haven't really converged the free energy landscape, how do we know that it's more converged? So this is only based on the extent of sampling, basically, but I don't have a full conformational ensemble that's converged to be sure about what I'm saying. It's, so it's mostly based on intuition and what happens early in the trajectories.
More questions for Lucie? I've got one. This is the maximum likelihood method. How does it handle the bias variance trade-off? Like you want to like smooth things out, you don't want to smooth them out too much that you erase features.
Yeah, so the maximum likelihood doesn't do this. It's more the cross-validation step that allows to do that.
Give it a minute, just a moment. It's a little bit slow sometimes. Any more questions for Lucie? All right, with that, let's thank Lucie again.
Oh, thank you.